From Microbe TV, this is Immune, episode number 58, recorded on August 1st, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. I cannot believe it's August.
Told you it was going to come before you knew it.
It's so fast.
And before you know it, you're going to be skiing again. Also joining us from somewhere in Ohio, Steph Langle.
Hi everyone. Good to see you. Yep, doing a little traveling in Ohio to see family and do some other things, which I'll get to follow up on, I think, in the next couple months. So good to see you guys.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone. I am trying to block out the fact that it's August and all of the things that need to get done soon. But it's much better to be here talking to y'all.
When does your class start?
The 29th.
Okay. Oh, mine's a week earlier than that. So
you still got a little bit of time.
I do.
Not enough time.
I know.
Well, summer is really only 3 months, and so it inevitably goes fast, right? You can't do anything about it.
Right, yeah.
When I say, when June hits, just start enjoying it, because if you're going to wait till August.
Yeah, well, July has just been, travel.
Yes.
So it's good to be home.
Absolutely.
And we all have some travel to do in October, right?
Yeah.
Yes.
Yeah.
That'll be great.
Which we'll tell you about another time, because right now we need to talk about science, and Steph is going to do the honors.
Yes, great. Well, I'm happy to be presenting a paper today. It's really interesting and kind of brings in human immunology. I think we present papers where we really try to dive in on mechanisms within mouse models, genetic mice models, or cell culture. But this paper, we're really going to tease out what is happening in the human intestine, and particularly during a disease called ulcerative colitis. And I'll talk a little bit about what that is, and feel free to chime in, if you have experiences with this disease or have taught it. But ulcerative colitis is one of two main diseases. Crohn's disease is the other one that falls into this umbrella of inflammatory bowel diseases. And IBD, inflammatory bowel disease, is an umbrella term. It describes these disorders that have this like relapsing, remitting, this waxing and waning, where you have it and then it subsides and it comes again. You don't know when it's going to come again. And it's very life altering, these diseases. It's defined by chronic inflammation of the intestinal tract, and they're not necessarily considered to be autoimmune diseases. Now, I think because we don't fully understand the mechanisms and they're so heterogeneous that it's not that they couldn't be autoimmune driven, but generally are not thought to be that. The real two main differences, and I thought it'd be fun to kind of pull up a couple slides, because we do have quite the YouTube following, that people that like to watch. So I'll describe the pictures as I share them. But the Crohn's and ulcerative colitis, it's where they happen that really makes the difference. So for Crohn's, it affects all layers of the GI tract. It's patchy. You can have areas of inflammation that are kind of interspersed with healthy, unaffected tissue. And again, it can present really anywhere along the GI tract. And in some cases, people have skin and joint issues. Mm-hmm. But Crohn's, it also has the diarrhea, abdominal pain, weight loss that ulcerative colitis can have. But ulcerative colitis is less diverse, and the inflammation is superficial in the layer of the intestine, and it's really limited to the colon. So the colon is your large intestine, and that's where ulcerative colitis really, if you're going to define it, that's the location. And again, causes sores, ulcer, bloody diarrhea. These are some of the most common symptoms. And, it's challenging because it can be difficult to get a diagnosis, and we really don't know what causes them. And when you look up, literature on this, these kind of Venn diagrams of what people think cause it, and there's some main ones. So genetics could play a role. They've done some genome-wide association studies and identified a number of genetic loci that are associated with IBD. But then there are environmental factors. It could be driven by the microbiota. It could not. So it's very complex. It's very challenging to pinpoint in these chronic complex diseases one cause, and likely there's not one cause, and it's probably multifactorial for many people.
So I think one thing I wanted to just point out, and maybe this is because I work with undergrads and this is something they get confused about, so maybe no one else is confused about this. But in case our listeners are, so Steph mentioned that this is not usually thought of as an autoimmune disease. That means that we don't usually think about there being some kind of self-antigen, that we're not seeing B cells or T cells attacking some specific aspect of self. That doesn't mean that excess immune responses or excess inflammation can't play a role.
Yes.
So you might think about it a little bit as an autoinflammatory disease. We definitely have the excess inflammation and excess immune response part that some people think of when they think of autoimmune diseases. But the key is there isn't a self-antigen.
Right. That we know of. And in the paper it's a great point, Brianne, because it's autoimmune diseases. We actually had mentioned in a previous show, we'd like to touch more on them because they are complicated and it's not always clear. So that's a great point. And in the paper, which we'll talk about, they do try to identify a self-antigen. It, in my opinion, didn't really pan out. That's probably not, at least in this cohort, we'll talk about driving it. But yeah, so it's complicated. And what you can't easily do with a complicated disease like this is model every specific aspect of it in a mouse. There are mouse models, and they kind of are divided into different categories for IBD. You can have chemically induced colitis, pathogen-induced colitis. You can do adaptive transfer of inflammatory T cell subsets, or you could have IL-10 knockout mice, which are IL-10 is a regulatory calming cytokine. So in papers where they're trying to get at mechanisms, they may present with these different mouse models. But what we don't really learn is that translational to humans? It's not always the case that our mouse models are going to be translational to humans. And so what this paper is attempting to do is to take some of the technologies that we have advanced, that we've really, we were able to get a lot of data at once and distill down what's going on. That's really what this paper is going to be presenting to us today.
And they're only able to do that because one of the treatments for IBD and Crohn's, or ulcerative colitis and Crohn's, is to actually take out part of the colon, right?
Right.
So I think if it gets bad enough, they actually remove the whole colon and just attach to the small intestine, basically.
Yes,
for ulcerative colitis, if it gets really bad. And amazingly, someone here had that done, and they said it kind of just regrows the colon, like it transforms partially back into colon, I guess, from where it is and the cues it gets. It's very interesting that the tissue changes. But yeah, so you're able to get sections of tissue because they do go through sometimes multiple surgeries.
Right.
Because otherwise we wouldn't be able to get access to any of the information, right?
And looking at the numbers in these cohorts, which I'll go through, there's a lot of people in these studies, and it seems that, which is why I think the data is presented. Some cohorts told us this subset of information with RNA-seq, but then they used this cohort to look at flow cytometry. And I think that's because they collected biopsies, which I'm assuming these individuals are getting regular colonoscopies, and you're taking small pieces out of the colon, and then you can use those small pieces for RNA-seq and some of these things. But these are small pieces. You cannot really, like what Cindy's saying, with taking a resection of the colon, you have a lot of samples. So what's presented here is, I think, mostly from colonic biopsies in these huge cohorts, which we can tell from the number of authors on this paper. These are the types of studies that require a lot of collaborators to get all of these samples. So I do want to mention, so Cindy brought up a good point with treatments. So before maybe you'd have to have a part of your colon removed, there are a sequence of treatments that you can undergo. So you would probably start with anti-inflammatory drugs, immunosuppressive, corticosteroids to try to calm down the immune system. Now there's something called infliximab or Humira, or is the drug name. These are anti-tumor necrosis factor alpha agents. So this is a monoclonal antibody that blocks TNF alpha, which is a pro-inflammatory, very damaging cytokine. But the issue is not everybody responds to infliximab or these anti-TNF agents. And that's likely, again, because this is a very heterogeneous disease and their pathology for the non-responders may not be driven by TNF alpha. It may be driven by something else, which we'll talk about in the paper. That really leads us, do we feel like we gave a good intro, everyone? Anything else others have?
Is there so there's another inflammatory disease, diverticulitis. It's not related at all to any of these, correct?
It is not my understanding that diverticulitis is considered an inflammatory bowel disease as it is described within the context of Crohn's and ulcerative colitis. I think because the difference being maybe it's these sores, these ulcers, in addition to the bloody diarrhea. Diverticulitis, only from family member having it, is like pockets. Yes. Somebody could write in, but I don't think it's a similar.
Yeah, they're pockets, evaginations of the colon, and then they periodically get inflamed because I don't know why. I guess they don't have a good.
I think they like trap little food particles that don't get fully digested, and then the bacteria grow in there and maybe produce gases and things that extend and damage. Another
solution to that is to remove parts of the colon as well. But I think one thing that's probably correct is that all of us know someone with some inflammatory bowel disease or diverticulitis. Yeah.
Yes,
it's very common.
It's very common, and it can be really devastating. And then particularly what struck me about IBD and Crohn's and ulcerative colitis is that the onset for a lot of people is like their late 20s. And these are individuals who've led healthy lives up until this point, and it's shocking to have somebody young and have this issue. So that really sets up what they're interested in looking at. So the title of the paper, this is ulcerative colitis, is characterized by a plasmablast skewed humoral immune response associated with disease activity. We have many authors. I don't see co-authors. So I'm going to say that the first author is Matthew Uzan, and the last author is Saurabh Mahandru. And let's scroll down, I believe from Mount Sinai. Somebody correct me if I'm wrong, because I have to scroll down very far to find that. I'll have somebody maybe look as I keep going to make sure I got the right association. But a lot of authors on this paper, and a couple of them that I noticed. So an immune alumni, Gabriel Victoria, you can find at the end, Gwen Randolph at Wash U. These are some really great.
One of her papers.
Yes. Yep. Luca Messin, he's the third, some really great B cell people. So you can see that this is a really heavy hitter B cell author list. And so you can imagine what some of the things we're going to be talking about. So there are a couple technologies that they use in this paper to be able to define what's happening in the intestines of these individuals. So two of those things, the first one I'll describe. So RNA-seq sequencing is you're going to be able to determine and take a look at all of the transcripts in individual single cells. Now that's different from bulk RNA sequencing, where you basically are looking at all the cells. You're not able to parse through. Somebody gave the example of like a smoothie. If you mix together a bunch of different fruits and you wanted to characterize what that is in the smoothie, that would be bulk RNA sequencing, but you're not going to be able to identify each individual type of fruit. Single cell sequencing allows you to do that. You can see which type of fruit is mixed in this population. And so if you think about RNA transcripts, you get a ton of data back from these type of analyses, and they require not only a lot of storage, but they require post-analysis types of algorithms to reduce the dimensionality of this data. So there's single cells plus all the transcripts within that cell. We have to find a way to organize that. And Vincent, I did send you the slides, and if you're able to find them, one of the slides is one of these maps that we look at, these U maps of all the different groupings that RNA sequencing data gives you back. And what's great about RNA sequencing that you can get this type of data from flow cytometry, which is looking at cell surface expression of proteins or internal proteins, where you can categorize them. You can look at the cells in different groups, but you can't further hone in on them deeper and deeper to further subcategorize and see, okay, well, I'm looking at plasma cells, but there's all these subgroups of plasma cells that are defined by these transcripts that are upregulated. Let's keep digging down and down. And so it allows you this higher level of specificity.
And it kind of flips it on its head, right? Because if you're doing flow, you have to choose your markers and you say, I'm going to look for this particular subset.
Right.
Whereas with single cell RNA-seq, you can say, okay, we know B cells express CD20 or CD19 or whatever it is, B220. And then you can say, okay, what else do they express and what's common and what's not? And so you can start to parse out new populations of cells that you couldn't see any other way or you couldn't distinguish any other way. But now that you have this fine map of the transcriptome, you can now define subsets of cells that you couldn't see before.
You can sort of let the cells tell you what the subsets are.
Exactly.
Yeah.
Exactly.
Right. Which brings us to, well, how do you deal, excuse me, my water bottle, with all that data is, and this is not my area of expertise. So I've done this type of work, but I give my samples to a core who create the libraries. They isolate the RNA, they create the libraries, they do the sequencing for me. And then I work with a biostatistician to help me bring this dimensionality down so I can get the clustering. And they do things called unsupervised and supervised clustering. And that can either be you kind of help the algorithm, you tell it how it should cluster based on predetermined groups of transcripts that you know are associated with T cells, B cells. There are a lot of things already out in the literature that we know define these cell types. Or you could do unsupervised clustering where you kind of loosen it a bit and you kind of let the transcripts tell you what should be these groupings. Both of these things were done in this paper. And so that's the technology. Now let's talk about the population they're looking at. So I brought up that there were 3 different cohorts. They have this primary cohort where they recruited individuals, both healthy controls or people who have ulcerative colitis. Then they have these other cohorts that they call validation cohorts. And we'll talk a little bit more later on about what those were used for. But the technicality of them is that for the primary cohort, you are recruiting them and you are doing the analysis after you have developed your hypotheses. But in the validation cohorts, they've already analyzed these people and a lot has actually been published. So you're retrospectively looking at them and saying, okay, is what we're seeing in the primary cohort, can we validate that in these other cohorts that have been published on? So the paper kicks off by defining very specifically single cell data in 4 healthy controls and 4 individuals with ulcerative colitis. So not huge numbers, and that's something we need to keep in mind, when we're looking at these data. But again, probably this type of work, you do need a lot of cells. And so they had the biopsies necessary, they had enough cells. Okay, now we can dig into what's going on with these tissues.
It's also kind of expensive to do on a ton of samples.
Oh, yeah.
Yeah. I have to budget for this soon. So I can't, what is, I don't
know the number. I just know that it's really expensive.
Like 25,000 to do a good solid experiment.
Right. Right. And you're going to be spending money on, you have a biostatistician core as well. Yeah. Right. Before that. Yeah. So that's the numbers. Now they talk about what type of tissue of these individuals they're looking at, and they describe as the lamina propria. Mm-hmm. And so if you're thinking, we're going to think about what an intestine looks like. You have these waves of villi, which actually the waves are more spread out in the colon. They're higher in the small intestine, they're more spread out. And these waves on the edges are these single cell enterocytes, these epithelial cells that respond with the environment. Below that single cell layer is the lamina propria, and that is where you're going to find the majority of immune cells, particularly effector cells, because the lymphoid tissue is kind of further down, the mesenteric lymphoid tissue. You do have some specialized lymphoid tissue called Peyer's patches, which we're not really going to touch upon, but that's where in the lymphoid tissue, as we know, you have naive cells, B cells, and T cells becoming educated. But the lamina propria is where these educated cells are moving to perform their effector functions. And so that's the tissue that they're dissociating. If you can envision, you have to get these single cells out of the tissue, so you have to use enzymes to dissociate the tissue to pull those out.
And that also kind of gets back to the point earlier about how difficult it is to get these samples, and that we are not going to necessarily have samples like this easily from any person.
Yes.
No.
Yes. That's right. And so, we will discuss because not a paper can't cover everything. There's going to be various gaps we'll discuss in this paper, but it should always be kind of remembering what Brianne just said, is that it is very challenging to get these samples and expensive. So go ahead.
If you want to tell me what slide so I can give you slide. That was your first one.
Yes. So right? What Vincent is showing, and for the audio listeners, is the different places in the colon where you have these different disease manifestations. So for Crohn's, it can be anywhere. And for ulcerative colitis, it's very specifically in the colon. So the next slide is these maps of single cell sequencing, and you can really see these different colors very nicely are showing you how the dimensionality is reduced, and you can then see where these cell subsets are located, and then right below that, we're furthering diving down into these different subsets to see, okay, what are the subsets of the subsets?
And sometimes when you look at this, it gets really confusing. But if you're looking at what's on the screen, there's a whole bunch of different colors, and each dot is representing one cell, right? And the distance that it is from the next dot is telling you how similar it is to that. So if there's clusters, that means that the cells have a lot in common. And if it's far away from another cluster, there are enough differences to separate them. And so conceptually, when you look at these things, sometimes it's hard to figure out exactly what it's showing you. But basically, in 2 dimensions, it's showing you how far apart these transcriptional profiles is. And so it's not one gene, it's all the genes.
Right.
And that's like really hard to wrap your head around sometimes.
Yes.
Because it's like a similarity score across the whole genome.
Okay, so next slide. So this is looking at the intestine. So you have on the left is the small intestine, on the right is the colon, and you can see what I described as the single enterocytes, the epithelial cells lining, and then what's right below that, where all of these, what they're describing as the plasma cells, that's the lamina propria, and that's what they're looking at. Okay, so maybe what is the next slide, just so that No. Okay. We'll get to that in a second. We're going to talk about B cell biology. So with first RNA sequencing, they're really looking broadly at all the different cell clusters in kind of this supervised way. They're not really seeing differences, but then they say, okay, let's dive into the B cells and plasma cells. Now, they don't tell us in the text, and maybe they didn't have enough text to tell us why, other than there's just a lot of B cell people on this paper, and it's just, this is an undefined area. Let's look into it. But they really just jump to, okay, we're going to look at B cells and plasma cells.
Yeah, I was going to ask that, is because why would you go and look at this? Because my understanding of what we know about, especially ulcerative colitis, is that there's connections with innate sensors. And so the precipitating event is there's some sort of gap in the epithelial layer, bacteria get across, and that's not always a bad thing. That happens all the time, but that the control of those bacteria, the detection of them, the control of them, and the level of inflammation are inappropriate, right? So you don't clear the bacteria, they grow, and so that triggers it more. Or the response is overzealous, right? And so there's an overabundance of inflammatory cytokines and cellular processes, which are bad in the intestine because we need to transfer nutrients, right? So we don't want to have a huge amount of inflammation in the intestine. And so those 2 things are what's wrong. And so I was trying to wrap my head around, well, why would they go after B cells? But I think the reason why is because there's also this component of microbiome that you were talking about. We think there might be different compositions of microbes in the intestine, and maybe they're inducing different levels of IgA antibacteria to control the bacteria. And so there's this crosstalk between the intestine producing antibacterial antibodies and the bacteria being controlled by that or not controlled by that. And so seeing whether there's differences in B cells or antibody production might give you a clue to whether that balance is out of whack.
Absolutely. And what might not be captured here, and we don't know as people in this field, but I learned by reading this paper, is that there was recently, I think in the past couple years, another group and another cohort had demonstrated that antimicrobial IgG was dominating the response in ulcerative colitis patients. And so it gets to this idea that it can be antigen specific, that there is an inappropriate response to your microbiota, and it's driven by IgG, which we'll discuss. There's some corollaries there. And so maybe their framing of, well, let's go into the B cells is because the field was moving in that direction. But we don't really get that nuance. So they then dive in and they are looking at different clusters of cells, and they can identify plasma cells, naive B cells, memory B cells. And I just want to describe, which is the slide that Vincent had pulled up, the last one is our different subsets of B cells and what's going on and where are they? And that's going to play a role in what we talk about. So we know that, I say we know, not everyone knows, so I'll just plainly describe it. You have a naive B cell in a secondary lymphoid organ that's going to, for the first time, be seeing antigen presented to it from something like a follicular dendritic cell. It's then going to interact with these T follicular helper cells that there's peptide presentation by MHC2 complexes, and the primed B cell is going to proliferate. And this is happening in a B cell follicle, and those cells can do different things. They can become germinal center independent memory B cells. So they can go off and be memory B cells. They can be germinal center B cells or short-lived plasma cells. But in this phase 1, they really haven't done a lot of talking in the germinal center. They haven't done this iterative presentation of the antigen, undergone many rounds of somatic hypermutation or these single point mutations to change their B cell receptor and just become better at binding that antigen. So short-lived plasma cells, they have a high cycling rate. If you look at the genes inside them, they are more terminally differentiated. They're proliferating more rapidly compared to B cells that kind of stick around in the germinal center because they need to become more mature. They're going to go back to the follicular dendritic cells. They're going to be represented by the antigen. And because they had been primed, they remember it, they bind more tightly to it. And that is a stronger signal to maturate. And this is usually correlates with more, again, somatic hypermutations to make sure you're fitting that antigen as best you can. And those cells are more likely to become long-lived memory B cells, long-lived plasma cells, because they have incurred those signals in the germinal center. So what we talk about in this paper really reflects these different phases. Your naive B cells, your short-lived plasmablasts that come from this first round of antigen presentation, or long-lived plasma cells that come from an appropriate maturation immune response in the germinal center.
And when you're saying germinal center, we're typically thinking about this in a lymph node, right?
Correct.
So
yes.
For the intestine, that would be the mesenteric lymph node. So we would anticipate
Correct.
The majority of those events would happen in the mesenteric lymph node.
Right. Or there can be some of this happening in the Peyer's patches, which are these very unique structures in the intestine. But yes, mesenteric lymph nodes. And then those cells, those plasma cells, memory cells, they go to the lamina propria, which is where you can see them. Now, what's interesting, and this is a nuance, and I just want to bring it up before we move on, as a activated B cell that is moving from the stage of primary B cell or naive B cell to plasma cell, they go in this in-between state called a plasmablast. Plasmablasts have not gotten the appropriate cues from the tissue to really fully differentiate and start pumping out antibodies as much as possible. That really happens when these cells kind of migrate to their sites, right? They're leaving the lymph node, they've been primed, but they need to go to their effector site. And once they set up shop, they get the cues from the tissue, the IL-10, TGF-beta for IgA, other cytokines, interferon gamma for IgG, potentially, what we'll talk about. So plasmablasts, you can think about them, they're kind of the teenagers. They are in the in-between state, and they traffic a lot. They have more rapid cycling transcripts. Turnover, thank you. So that's the difference between them. I realized that I plugged my computer into a defective outlet. Yeah. So I'm nervous that this is going to die on me. So how about you guys talk amongst yourselves while I fix this problem? And I'll be right back. Anything about B cells that you that I missed?
So some of those B cells that are sort of germinal center independent are germinal center independent because they maybe don't interact with T cells in the same kind of way. They are responding to T-independent antigens. But some of them are also associated with aberrant immune responses and sort of autoimmune types of B cells. And so, the classic way that we think of B cells is going through this germinal center response if they are a T-dependent B cell. I think that it's also kind of key to point out that sometimes if there are some of these germinal center independent cells, they perhaps may be associated with pathology, at least in some other systems.
Perfect.
And the blasts, I kind of thought of it as you need to move the cells into one part and let them proliferate, and then some of them go back and they test their antigen again and they see if they rearrange something. Then they go back over and they proliferate some more and they go back and forth so that you can constantly be making more of whatever you have, and then you test and you can tweak and refine that. Does that also fit with what you're saying here?
I think so. Yes, I would agree with that. I think the plasmablast can be cells that are moving in between the compartments, or they can be cells that have exited for whatever reason. When they didn't receive the signal, but they received the first signal, not the second, and now they're moving outside the body. I have some thoughts on that, which will be more relevant later. So yeah, I think all those points are great, and I have solved my computer charging problem.
They say in the introduction, most of these antibody secreting cells are IgA producers, right?
Yes,
yes. There are fewer IgG producers. And so all of the things you just talked about, the interaction of antigen and so forth, would end up giving rise to more IgA producing cells, including memory, right, than IgG?
Yes, and that's a great point, and also a good thing to bring up for this paper is that the reason why the majority of your B cells become IgA producing cells in the intestine. And actually, most of these cells, the IgA producing cells in your entire body are found in the intestine, is because they class switch to IgA by two main cytokines. Those are TGF-beta and IL-10, which are produced by stromal cells in the intestine, but also other immune cells. And so the intestine is chock full of these things unless it is in an inflamed state. And then it is not going to produce those as well. And then you might have class switching to something like IgG, which in the context of what they find with their single cell sequencing is they do see a lot of IgA producing plasma cells. So they do see that, but the proportion of cells that are producing IgG are greater in the inflamed UC, ulcerative colitis, biopsies, significantly greater compared to the controls. And that extends to multiple different clusters they looked at, that it was a consistent finding that this was an IgG dominated response. Which
so it sounds like the B cells are getting the wrong cytokine cues to class switch to the wrong isotype.
Absolutely. And if you think of the baseline being IgA, there's a reason evolutionarily that we have IgA, because it is more likely to maintain the microbiota in a homeostatic, non-inflammatory state. If you have inflammation, as these patients experience, it's very challenging to digest food, to interact with the microbiota in a healthy way. So there's a reason that we don't have a lot of IgG. And so, as Brianne mentioned, they're likely getting the wrong signals, which means that they respond to those wrong signals by further producing that same wrong signal, which in this case they describe as interferon gamma. We think of interferon, we as in people who work with viruses, but anyone who works in innate immunity, interferons can be potently inflammatory but can be produced by IgG plasma cells and contribute to virus protection against pathogens like viruses, but also play a role in autoimmune diseases like lupus. So when they then see this phenotype, they then continue to use their single cell sequencing. You can process the data in particular ways. And so the way you look at the data, you can do many different types of comparisons. And they describe that for us. They call these things Euclidean distances, which is basically just saying what are the similarities between the cell types, and then how do those similarities within treatment group compare to the similarities amongst treatment groups. And they tell us that ulcerative colitis biopsies are just it's something that's very different there. And so then let's keep diving in. They describe to us this molecular signature database, which is just a way to take your single cell sequencing data and kind of cross-reference it amongst other gene sets that people have curated in the past. And what pops up? Interferon alpha, interferon gamma responses in those plasma cells. So it really, in this first part of the paper, is describing to us that ulcerative colitis is being defined, at least in these patients, as this acute plasma cell response towards IgG. And they also discuss a little bit about the naive population, which again is the beginning of the story for B cells, but they too had higher levels of interferon. Okay. So now that we know that, they wanted to take another technology. So we talked about flow cytometry, and we can label these cells and get similar, you can get similar types of data. You might not get as much data, but it agrees with the single cell data that when you look at the ratio of plasma cells producing IgG versus IgA, it's higher in IgG. And it's like this systemic like B cell response. So that using those two technologies, they were able to conclude that. But now I think they wanted to dig a little deeper into what's actually genetically going on with these cells. Now, it's kind of funny. I don't know if you guys agree, but in the first paragraph of this next section, where we're going to look at the genetics, we're going to sequence single B cells. They kind of tell us how they sort them. So they wanted to look at these short-lived plasma cells, right? The ones this is like the first responders. And they also wanted to look at peripheral blood cells that had seen the intestine before. And I'll talk about what that means. What marker are they looking at when you can say, oh, that cell has seen the intestine? But they kind of describe how they single cell sorted, and they tell us how many clones they were, but they didn't really say, and then we sequenced them. Okay? There's kind of like a gap in the description. If you weren't familiar with it, you'd be like, what? What are you doing?
Yeah.
Yeah. Okay, so they're taking these short-lived plasma cells and then these plasma blasts from the blood. And now I brought up, how can you tell that something has seen the intestine? There's a marker called beta-7. Beta-7 is highly expressed on cells in the intestine, and the reason that it's there, it's an integrin. And if it binds its cognate receptor, it's in context of the intestine, it's called MADCAM. It's a pretty cool name. It will help physically traffic those cells and keep them in the intestine. Now, if you find a beta-7 positive plasma blast in your blood, you're going to say, well, something's happening in the gut. Well, two things you could say. It could just be that these cells migrate in your blood normally, which they do, low levels of them. But if you see a consistently higher level, it can mean that there are some perturbations going on in your intestine, that something that was supposed to kind of stick in the intestine, this beta-7 integrin binding, is now in the blood more.
Well, and you also mentioned sort of this idea of there being some systemic change as well as the local change. And this also gets to the idea of, the problem in the intestine is kind of spilling out to be a problem elsewhere as well.
Yeah.
And also, if they're being activated in mesenteric lymph node or wherever, and there's more of them, you might detect more in the blood as well, right?
Right.
Because what I was thinking of, I often teach this when I'm thinking about it, if you have a Peyer's patch in the gut, why don't the cells just leave there and go populate the gut? And when you think about the size of the gut, it's like you're trying to move a cell through a bunch of tissue and try to find it. Whereas all of the villi and crypts and things are bathed in the vasculature. And so it's actually faster to take your immune cell, put it back in the blood and let it float around and make it back to the intestine so you can populate the intestine. That's my thought. Do you guys agree with that?
Yes.
I would. Yes. And I think that speaks to why we would have normally in our body cells that have been in the intestine or seen the intestine, just trafficking. And I think that there was a paper that looked at 80% of the IgA producing plasma cells that you would pull from blood are gut tropic, that they came from the gut. So you're right. We would normally find them there. And when we see an increase over baseline, and that's where I guess the baseline is important, it then could signal something is happening. So when I infected my pregnant and lactating pigs in my PhD with an enteric coronavirus, I saw at 7 to 10 days an increase in beta 7 positive PEDV specific cells that had come from the intestine, and then those numbers would kind of go back to baseline. So it suggests that with these ulcerative colitis patients that either you're just making more of those cells, like Cindy said, or that the disruptions in the actual tissue are I don't want to use the word leaky gut because I think it's totally overused and has, I don't know what that means, but you could have cells that are moving into blood at a greater rate. Okay. So let's then dive into what they found. So they did find a higher proportion of beta cell positive plasmablasts were IgG, again, following our theme in UC, in ulcerative colitis patients. And what's noteworthy is they say that they're IgG1. There are 4 subclasses of IgG antibody, 1, 2, 3, and 4. IgG1 is the most dominant, at least in the blood, and it is associated with inflammatory responses, and that can be really it's really driven by the FC, the stock portion of the antibody, which is something we'll get to chat about. But antibodies, if you hold up I love doing this, your Y-shaped with your hands, you can bind the antigen with the tops of your fingers, but the bottom portion still has a function, and that function can really drive inflammation. So if you have higher levels of that in blood or in the intestine, you can imagine there might be some effect or consequences. Okay, so now we're going to get into a little bit of genetics. So, which I would say that this was probably the least fulfilling part of this paper. I think it leaves a lot of questions that can be future papers. So they asked the question, what are the dominant genes that are being expressed in these cells? You pull out single B cells, plasma blasts, what genes are being used? Because what we know is that there's a number of functional gene segments in the human antibody loci, and they're looking specifically at the heavy chain. There's 3 segments, the variable, the diversity, and the joining segments, and they have different numbers, and they recombine in different ways to give you this immense diversity that our B cell receptors have. And what they say is that there's this one joining segment that was used more in the ulcerative colitis patients, and that was JH6. Now, they really just kind of leave it at that. They don't really give us any insight in that or the discussion, what the heck does that mean? But I can tell you from previous literature looking that naive B cells are more utilizing this JH6, but something like VH4 is more utilized in affinity matured B cells. It is starting kind of this story for us that not only are these cells more IgG, but they're probably less mature. They are less affinity mature. They spent less time in the germinal center. So that's clue 1. Now, what's the next clue? They talk about something called CDR3 length. The CDR3 is a complementary determining region. And on your antibody, holding up my Y, you have 3 of those per arm. So in each of your arm, there's CDR1, 2, and 3, and that makes up the paratope that binds to your epitope. So technically you have 6, right? Because in a dimeric IgA, then you would have 12. So CDR3 is kind of the one that it's the CDR that has this flexibility, and it changes in size, and we can interpret the length of it. It might mean something. And here they describe that in ulcerative colitis patients, it's longer. And again, they just leave us with that. They say it is longer, and here you go. But what does that mean? Well, in literature has demonstrated that the more affinity matured a B cell is, the shorter that gets. So in UC patients, if it's longer, again, these cells are getting cues to maybe exit the germinal center too early. They're not utilizing genes that might be beneficial to them, like VH4, and they're not shortening their CDR3 length. Little clues into what's going on. Okay. So they also were wanting to know how many clones can they find in these individuals? How similar are the cells that are circulating? Maybe that would give us some indication as to what's going on with these single B cells. So what they found is that there were 6.5% of the clones belonged to a set of expanded clones. So they looked for clones that were found at least twice. Like if I take your blood and I find 2 different clones, that mean that one clone expanded enough that you would find 2 in a randomly in a sample. So 6.5% in healthy patients, but 11.2% in patients with ulcerative colitis. So what, to me, and I'd like your feedback, what this suggests to me is that you have this hyperactive production of immature plasmablasts that have higher turnover, that are escaping potentially the colon. They're not sticking around in the germinal center to engage with their normal processes. And again, shaping this idea that this is a proportion of plasmablasts that they're skewed in they're not being allowed to mature.
So the longer CDR3 part, I don't know as much about that one. And so that one doesn't fit in my mind the way all of the other parts fit together. But in general, when I look at this, I'm seeing, okay, so there are some cells that are not going through normal developmental processes. They have markers of kind of having not completed as much development as they usually would. And there are too many of them, which means that they probably were dividing a lot as a result of this not doing their developmental process. And so it does look like maybe there are some cells that are escaping early, having not had the proper activation or proper education and in these patients. So it kind of tells me that this is a little bit of an issue early on in the development of those B cells, that somehow something went wrong in their developmental process to make them leave the germinal center, as Steph said, and potentially cause inappropriate responses.
The one thing that I'm having trouble wrapping my head around is that I thought that the more focused and refined the response is, the more oligoclonal or monoclonal. Whereas if it were activated really early, I would anticipate a broader response. But that's not what they're seeing, right? They're seeing very few clones as opposed to a broad response. So how do you fit those two pieces together?
Yeah, so I would say that I think what we don't know is the antigen specificity. So I think it's challenging to really hone in on what those expanded clones are. And that would probably answer our question, which is why I think they try to answer that in the next paragraph. Because it may be that those expanded clones are either broad or specific based on their specificity. That yeah.
Okay, so they may be more broad reactive.
Right.
So not so highly specific and refined.
Right.
Right, because they don't have as many somatic mutations.
Correct.
Right. And I'm sorry for not mentioning that, which is a key point in this. They do not have as many somatic hypermutations, which fits within this story. So I think what we're learning is that, right, they're not sticking around in the germinal center and they're getting cues to proliferate inappropriately. So I don't know if it tells us anything about the ability to bind to their cognate antigens either strongly or loosely. Mm-hmm. It's more telling us the story of their proliferation.
Yeah.
So they do, yes. So they do try to answer that. And it's one paragraph. This really, I think, is what everyone would like to know. What are the antigen specificities of these pathogenic interferon gamma IgG1 skewed B cell responses? Plasma blasts, right? Like not plasma cell, they're too early. They run these antibodies through a panel of microbial antigens, a multiplexed assay. They tell us there's like 3 microbial antigens that their ulcerative colitis patients bind to. They don't really tell us any more about that. I looked up what those were. There's one called Enterobacter cloacae and another one called Lactospira. You can tell I'm not in the microbiota field. I can't pronounce any of these, but it doesn't give us much information into what these are binding to. Maybe like some other literature which they cite that did show this very IgG microbiota directed. They are not necessarily seeing that here. So then they explored the idea, well, could this be an endogenous autoantigen? This was also based on recently published work that alpha V beta 6. I remember I brought up the integrin beta 7. Alpha V beta 6 is another integrin that's helps cells move or stromal cells connect. It's on the surface of these cells that you're responding against that, and your B cells are pathogenic against this. They pull out antibodies from single cells and they say, do you react to this integrin? And only one clone from one patient. And they didn't look at many people, right? I think there's only 8 total. But it would not suggest, at least in this cohort, that story is true here.
Didn't seem to have done the multiplexed assay with healthy control, right?
Right. So that would be helpful, but I wonder if they didn't, because they didn't really see much of a response in the UC patients. I'm not sure. There's only 3 hits.
Is that surprising that only 3 antigens are recognized by all these antibodies?
It is well. So it's interesting in the field. So in this field of antibody-directed microbial responses, that Andrew McPherson, who's a really great mucosal immunologist in some Nordic country that I can't remember, he had demonstrated that the IgA response against microbiota was specific. That for a long time we thought it was like polyclonal, but that it actually is a monoclonal response. And so I am a little surprised that we don't see more IgG directed against microbiota. But regardless, we move on and we're moving on to really, okay, well, what would cause this inflammation based on the results that they're seeing? So we remember that it's IgG dominated, IgG1, which Vincent, if you wouldn't mind pulling up the next slide, because this is a slide describing what the FC portion, the bottom stalk portion of the antibody can do when it binds FC gamma receptors on the surface of innate immune cells like a macrophage. You can have IgG that binds to a pathogen on the top of the Y and then locks in on the FC gamma receptor in the macrophage to induce phagocytosis. But you can also have antibodies that are kind of complexed together with antigen on their Fab region, but they're all like binding the same antigen, creating this like glob, this mess of IgG immune complexes, and that can also stimulate macrophages to secrete their inflammatory mediators. So what I would have loved is that you could pull out the B cells from these patients. You can look at the genetic sequences of the antibodies. You recreate those antibodies recombinantly in the lab, and you do something like an antibody-dependent phagocytosis assay to ask the question, do ulcerative colitis patients have more of these antibodies from their B cells that induce phagocytosis? I'm assuming they couldn't do that because of lack of sample, as we've discussed before. So they did something a little differently. They took immune complexes and they added them to murine mouse colonic macrophages. And then they single cell sequenced those. And what do the inflammatory phenotype look like when you take IgG immune complexes and you add them to macrophages, what does that look like? They also did LPS stimulation of murine colonic macrophages. Somebody could speculate why. I guess it's just like another inflammatory, a positive control, I guess.
Yeah.
And they compared them. They compared these phenotypic inflammatory marker transcriptomics to the inflamed intestine. They found that ulcerative colitis patients look more like these murine macrophages when they're induced. And so they make the connection that this could be the reason why you have this inflammatory response happening in the gut of these patients is these IgG immune complexes. But they also saw the same upregulation with LPS. So to me, it wasn't exactly, it's not going to be confirmatory. We would need to do more work, I think.
Yeah, I think part of why they wanted to look at LPS is because when you have those breaches in the barrier in the gut, LPS is going to leak across. So those macrophages are going to be seeing both of those things together or in similar ways. And they wanted to know whether those immune complexes were inducing an inflammatory signature. So you can compare that to LPS to see. But it really is different because the TLR4 is what's going to signal to LPS. And so that's going to induce a specific program of response.
Right. So it's trying to get at this question of what is causing this. This is just like the big question in this field. Could it be IgG immune complexes? Sure. But what stimulated the IgG immune complexes? What is the antigen? So still leaves us with questions. They use more single cell RNA sequencing to look at. They find resident-like macrophages that only in the ulcerative colitis have inflamed the associated genes upregulated that look like the LPS. So again, that this could be the reason that this happens.
So what we're saying so far is there's something that's triggering this dysregulation of the B cells. So you're getting more production of the IgGs that could then form immune complexes when the barrier is breached. And then those can trigger the macrophages and then they initiate an inflammatory profile, which then precipitates the whole inflammatory response.
Yes. Yep. Absolutely.
And that's why it's refractory to antibodies to TNF because it doesn't involve TNF, that mechanism.
Yes.
Right?
Yes. Great point. Yes. Okay. So they then want to ask the question, well, it's not just B cells, right? All of these things are acting in concert. And we talked about that the B cells are producing interferon gamma in response to something. So what is the something? And so they dive into T cells and they do colonic T cell transcriptomics to look at the global composition between ulcerative colitis and healthy controls. And they see things like expression of Th17, which is associated with this disease with inflammation, Th1 effector cytokines, like interferon gamma, things we've been talking about, that they describe as contributing. They look at some of the significant differences to say that, okay, these are interdependent, which I would say, like, of course, I mean, as the immune system is interdependent, that the production of interferon gamma that they're seeing in the T cell subsets could then stimulate production of interferon gamma in these IgG skewed plasmablasts. But they specifically want to bring our attention to something called CXCL13 positive TPH cells. Now, okay, TPH cells are T follicular cell-like peripheral helper cells. So this is kind of interesting to think about. So this is a unique subset of T cells that has been found in the periphery in individuals with autoimmune diseases that they say has not been found in ulcerative colitis patients, which they're finding here, that they have this B cell attracting chemokine CXCL13 that they produce. But they also produce some T regulatory types transcripts, but that these T follicular-like peripheral cells are higher in the inflamed ulcerative colitis patients. And so are they contributing to either helping recruit B cells to promote interferon gamma production? They look at immunofluorescence and see these CXCL13 positive TPH cells at kind of like the expansion of the inflamed portion, just distal to where the pathology is. And they say, okay, well, they're near, but in the ulcerative colitis patients, but not in the controls. So, to me, this section was a little challenging to interpret other than they're there. This is the first time they're being described in ulcerative colitis, but do they really play a role? It's unclear.
Yeah. So
would that be fair about this section?
Absolutely.
I thought that one of the things they are trying to do is figure out who's making the interferon gamma that might be driving the class switch to IgG, right? And so these cells are unique because they're not normally found, or they haven't been previously described in the intestine, and they're expanded. These TPH cells are expanded, and they have an interferon gamma production signature, and so it's like implicating them in that initial process.
Yes.
So my question is then, are the B cells are probably switching then once they get to the intestine and interact with these cells? Is that how you would interpret that?
That's interesting. Because they describe them as being these peripheral cells, but then they also find them in the intestine. So I don't have a good sense of like their spatial distribution and like where, at what point are they interacting? Are they in the lymph nodes at all? I agree. I think that they are trying to hone in on that, but I'm not really clear at what point they maybe would be interacting.
Yeah.
And maybe they're just bringing this up also because, okay, this is now something maybe we can dig into with a mouse model.
Right. So
it's useful to repeat their summary. They say, we find evidence of a population of TPH cells with potential to support recruitment of naive B cells together with activated T cells like interferon gamma could drive expansion of a subset of interferon gamma responsive naive B cells and promote IgG class switching in ulcerative colitis, right? So that's what they're thinking is happening.
Yeah, that's a great summary. And also points to, they're saying that because those cells are not there in the healthy controls. Because there could be other cells that help drive this, but it's the fact that they're not there in the healthy controls.
Yeah, so I kind of took this as...
Great. So
it's still hard, though, with this kind of analysis to figure out chicken and egg, right?
It's so hard.
Because it's easier to do that in a mouse model because you can knock something out and you can say, does something else happen? But with the human system, we just kind of, it's all correlation, right? Yeah. But it's building towards that story that would suggest that their model is correct.
Yes, absolutely. And I think with these type of human immunology cohort, small sample size, you are trying to include redundancy in your studies using different technologies to try to get at things that you don't have a reduction model. You can't remove a cell and see what happens.
Right.
Okay, so we're kind of winding towards the end of the paper. I think broadly we understand, what is happening in regards to the major themes that we're seeing skew towards interferon gamma IgG that's being stimulated by something outside, maybe the CXCL13 positive cells. And then they ask questions about the circulating gut homing plasmablasts and plasma cells, and that are they increased in our ulcerative colitis patients? And they use beta-7 as a marker, but they also look at other tissue homing molecules. There's many. And they describe an increase in beta-7, a decrease in beta-1, which beta-1 all these integrins are heterodimers and they pair with something else. So like alpha-4 beta-7 or alpha, I forget, 1 beta-1. I can't remember what the other beta-1s are, but they say that the beta-1 is decreased, but then CXCR3 is increased. They liked CXCR3 because it is a receptor for interferon gamma induced ligands. And these two ligands or chemokines are expressed by macrophages. So that model would be you have these two chemokines, these ligands, CXCL9 and 10, being produced by inflammatory macrophages, which they show are expanded in the UC, in the ulcerative colitis, that they then bind to interferon gamma. They're produced by interferon gamma, and that contributes more to this inflammation, driving pathogenesis. So I don't, they're using, and what's helpful in this last part is they want to use their validation cohorts. I brought those up in the beginning. These are cohorts that were previously recruited. So they're looking at the hypothesis in reverse. They're asking questions from something that's already been collected because they want to validate something called a biomarker, which can be very elusive and challenging to hone in on something, because basically you want to say, if I find this thing in someone's blood or tissue, is it indicative of disease later on? And they ask that question for beta 7 expression on plasma blasts. And they say that yes, when adjusted for things like age and sex and disease extent, that about a year and a half later, we can say that the levels of beta 7 plasma blasts are associated or lead to an increased probability of disease complications in their colitis patients. Lastly, which is, I think they put in there as another potential biomarker, but it's fun to talk about, is something called they say IgJ. It's the joining chain. It's in immunoglobulin J. This protein, when expressed, will help bring together our secretory antibodies like IgA and IgM. And what's really interesting about the joining chain, this J protein, is that it is expressed in all plasma blasts. So whether it dimerizes with secretory antibodies, that's a function of whether that plasma blast class switches to IgA or IgM. But you can find it in IgG plasma blasts too. It's higher in plasma blasts, again, because I think that those cells are undergoing, they're trying to get to their terminal plasma cell state. And they try to say, is increased expression of the joining chain associated with rectal bleeding scores? And they found that. So it could be another marker. So a plasma blast marker, biomarker.
So I have a question, because one, I had to look it up, because I know it is J chain, and I was like, what is IgJ? Because I thought IgJ was made by another species of animal, and because you have IgY and all of this, and I thought, okay, so then I got IgJ. Okay, I got it. It's a J chain. But what you said was that all plasma blasts will make it. And so I'm curious. So why do cells that are not making multimeric immunoglobulin, like IgA or IgM, why would they make J chain? Does it have other functions?
So it's really understudied. J chain, we have crappy antibodies. The structure was recent. I don't even know if we have a great structure of it. We don't know a lot, because it's a great point. Why would the cell take the time and energy to make this protein in a plasma blast or plasma cell that's not going to dimerize, especially if it's already class switch? It's not going to make IgG and IgA. So I am unaware of a benefit of expressing J chain in a cell that's not going to use it.
Because it happens. I know. If you think about it, if you're going to design the system, you'd have cells make it normally because they're going to be making IgM, right, to start. Mm-hmm, then if they get induced to class switch, they could shut off lots of other things. Why couldn't they just shut off expression of J chain, right? And then if they're being class switched to IgA, whatever the signals are that are inducing IgA class switch could maintain expression. Cells are sophisticated enough to do this. And if they're not doing it, there's probably a reason.
Yes, and I would love to learn that reason. J chain is a very cool protein. It's only 15 kilodalton. It connects our secretory antibodies to make them more stable at the surface. But I'm not really sure that answer. It's a good question, though.
Huh. Interesting.
So that's how they round up the paper with looking for biomarkers. So I think what we learned from this paper is that there is definitely something going on with the plasma blasts. They are skewed towards IgG. They're pro-inflammatory. Could they be driving IgG complex mediated activation of inflammatory macrophages? Sure, that could be the reason. But I think this paper contributes really to our understanding of the disease. And so then we can further understand what causes then this to happen.
And I wonder, because there are subsets of these individuals, right? So probably some have these expanded IgGs, maybe some don't. I wonder if the ones that do may have more of those other signs symptoms of disease, like the involvement of the tissue or whatever, because they might have immune complex mediated autoinflammation.
Yeah, I
Do you
agree.
know if
Like,
that's true?
I would imagine that what you said is true, that on the spectrum of these diseases, as they see many different reasons, that there's probably a subset of IgG immune complex patients. And maybe that's what we're capturing here. Maybe these cohorts there's a lot of them. And, it's hard to know with only, because they did a lot of this work with only 4 ulcerative colitis patients, if this is universal or is this a function of, just this population. So, yeah.
But that could be something that you could tease
unlikely
out.
to be universal with only 4, right?
Yes. Yes. Agree.
But assuming this skewed B cell population is the cause, right? Which we don't know. As you guys said, it could be a consequence.
Right. Chicken or egg. Yeah.
You said, Steph, figuring out why. Like, what's the mechanism for this skewing? That's really the key, right? What is going on?
Right. Right.
Because eventually you want to figure out how to treat it, and you need to know that, I guess, right?
Yeah. Yeah. And they do bring up, how would you use this information to better treat patients? Could you block some upstream cytokine that the macrophages are responding to, that then they produce more interferon gamma, which are all hard questions because all these cytokines have other roles, right, in your body. But for sure, only targeting TNF is not going to get us there.
Right.
In theory,
I mean, is it possible that it's in part dysbiosis that leads to? So we have 3 microbial antigens that predominate in the IgG response. So maybe that is a consequence of dysbiotic intestines, which is really the key. Who knows, right?
Yes.
In which case, then maybe probiotics could help. I'm sure people have looked at that, right?
Well, and all of these things could be helped.
Probiotics targeted, is there a proliferation of another niche that needs to be, reduced? It is very challenging. I think without knowing more about the microbial binding, they really just didn't see as much as some other papers. It's challenging.
And one thing is they have this huge set of data now, the single cell RNA sequencing data, and they really only looked at the B cells. But it would be interesting to look at what is going on with the transcriptomes of the other cell types. And
well, they sort of look at the other cell types.
Because I even think, and I'm going to have to scroll down to try to find this figure. In one of the figures, there was a big change in the CD4 T cells. Am I correct?
I think so. Yes.
And I was like, oh, well, I wonder why they didn't look at that. That's the proportion. It was like the proportion amongst the
fraction of T cells, the T resident memory cells, CD4 T resident memory cells were massively expanded in their patients that they tested.
It felt like...
I know you can't do everything on paper, but if there's a follow-up T cell paper, I would assume that you could dig into that. Okay, great. Any other final comments on our human immunology clinical cohort paper?
This paper illustrates why immunology is difficult, because if you don't know what B7 is, you're going to have no clue what's going on, right? Unless you go and look it up, and in which case it will take you 2 weeks to read the paper.
Right. Or like that one line where they say the length of CDR3 was longer. It's like, okay, well, what does that mean? You go to the discussion, it's not there. So it is very challenging, and hopefully helped work us through that. Well,
it's what we're trying to do on this podcast, right? Is have experts like you guys and explain things that you would normally take a long time. I have so many students who come up and say, you saved me a lot of time. I don't have to read the paper, which, there's no substitute for reading a paper and decoding it on your own. But if you want to do a lot, that's where we can help.
We may be missing Brienne. I think she's frozen on my screen.
She is frozen on my screen too, and I think she can hear us, and I think she's frustrated because she's like, "Can you not hear me? I'm talking." So
I'm sorry, Brienne. We missed your gems.
Yeah, for those of you who are wondering why Brienne is so quiet, I think she's just frozen.
Well, it may be that her recording. Hey, Brienne, say hello, everyone. I want to see if we see a
blip.
Hello, everyone.
Blip here. I see a blip. Okay, so you're recording. So just
talk.
Unfortunately, we don't know when you're talking, but it's too bad. I was going to ask you to read one of the letters. We cocould stop and restart if you guys want, or we can just wrap it up.
That's up to you if you want to fix the recording.
I think it could be fun to do an email episode in the future, because we have some cool, we have our interview coming up and we can have an email episode. I think I want Brienne here. So I think. Well,
we only have 3 emails, right?
Oh, I didn't.
Well, these 3, okay. And then we have a bunch of emails from people who were suggesting autoimmune topics, right? Because we brought that up, but I didn't put them in because it's not something you can answer. It's just do you want to do this paper? Do you want to have this person on the program? That sort of thing.
Yeah.
Brienne seems to have left the show notes now. I don't know.
Well, we can wrap up so that we can make sure to have Brienne.
Now she's back.
Okay. Oh, she's back.
I hope my stuff's working, because I'm not actually seeing my blipping on my screen, but you all hear me, so I guess
I see your blipping, yeah.
Okay.
All right, well then let's wrap it up and we'll save the emails. The next time we record will be in Ithaca, right, Cindy? Yeah.
Yep. And are they going to join us on August 17th at 10 a.m.?
I thought you guys thought you could join us by Zoom or Zencastr, right, Steph?
Yes. So that's totally fine. Yeah.
And Brienne has it on her calendar. Good.
It's on my calendar. Yeah. So I'll be up there with Cindy and our guest, and it should work. All right. That's Immune number 58. You can find the show notes at microbe.tv/immune. Questions and comments to immune@microbe.tv. And if you like what we do, please consider supporting us. We need your support to do this. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thank you. I learned a lot about B cells today. Really great.
Yeah, I always learn a lot. Yeah. Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thanks. Thanks for letting me talk about the paper.
You don't have to thank us. We're happy to have you.
You know me. I'm just polite.
Yes, of course. Brienne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brienne.
Thanks. It was great to be here in the amount that I was here.
Thanks. I had a lot of fun. Or something like that. I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back in a couple of weeks.